from where we left, which is the effect of diffusion in the fungal pellet. And if we go to the slide, what you'll see is that we wrote this equation, if you remember, which is the diffusion equation. So on your left hand side is the diffusion of oxygen, and on your right hand side is the reaction that occurs because of the growth of the fungal pellet. So the fungal pellet essentially has to grow. What was the major assumption out here? The major assumption was of pseudo steady state approximation. So the idea was that though the fungal pellet is actually growing in radius, but the rate of growth of the radius is much smaller than the rate of diffusion of oxygen. As a result, and how did we conclude this? We concluded this through time scale analysis. We measured what is the time scale of the growth of the radius of the fungus, and we measured what is the time scale for diffusion of oxygen in the fungal pellet, and what we found was that the time scale for the growth of the radius is much larger than the time scale for the growth of the fungal the diffusion of the oxygen, or in other words, the diffusion of oxygen is a much faster process as compared to the actual growth of the radius of the fungal pellet. So as a result of which we can write the pseudo steady state approximation, or in other words, we can assume that the diffusion of oxygen is a steady state process as compared to the growth of the radius. Fine. So based on that, we write this equation out here, and what you see over here is that the temporal term in oxygen concentration is missing because of the pseudo steady state approximation. Fine. So this is the radial diffusion. Excuse me. This is the radial diffusion, and this is the reaction term, and this is written in dimensionless coordinate. All of it is written in dimensionless coordinate, and I'm not redefining these dimensionless coordinates. The reason I'm not redefining is that we already did this in the immobilized enzyme case. The last example we did in the immobilized enzyme already had the same variables and same dimensionless coordinates. So you can go back and look up for that. So I'm not redefining it. Now, the assumption here is that oxygen is much greater than Km, which is a very reasonable assumption, because if you want to grow something, or fungus is growing, for example, oxygen is typically in abundance as compared to the Km. So that's a very straightforward assumption. Now, when you make that assumption, what you find is that this equation, which is a Michaelis-Menten kinetics, reduces to a first order kinetics over here. Okay, is it clear? So the Michaelis-Menten kinetics over here reduces to a zeroth order. Reduces to a zeroth order kinetics over here, because when CO is much larger than Km, then what happens? CO in the numerator and denominator cancel out, and this is what you have. So this is what we got over here, and S naught is all of this combined, the Km and beta and so on combined. So R square, R is the radius of the mold, of the fungal mold at that point of time. Okay, is it very clear? R, ideally R is a function of time, it's increasing with time, but since we are doing a pseudo steady state approximation, so R over here is the radius of the fungal at that point of time. Fine. So we write this equation, then this is a very straightforward zeroth order on the right hand side, and you can integrate it straight away. And what are my boundary conditions? The one written on the screen, which is that the oxygen concentration at the outside radius, R equals 1 or R equals big R, whatever it is 1, and DDR of CO naught is 0. Okay, so let me show you this, why we make this assumption. So this is my cylindrical mold. My reaction equation over here is 1 over R DDR of R. New max I used instead of R max, the reason being that R now I'm using for the overall radius of the mold, that's why I changed the effect. S naught is that Km over beta, that you Km times beta, sorry. So this is my equation. The boundary condition inside is, so this is my R, this is R equals 0, this is R equals 1. So R equals 0, my boundary condition is symmetry, right? So equals 0, R equals 1. I'm saying that oxygen is present in a certain concentration. Okay, so CO2 equals 1 in the dimensionless coordinate, which means that actual value of concentration is something, some value CO naught or something like that. So CO2 is equal to CO naught in dimensional. When you turn into dimensionless, this is what you get. Right? So this is the boundary condition we have. We can straight away integrate this. This is a straightforward integration, and we can get the profile of concentration within the mold. So why are we doing this? And the reason would be obvious. One of you I think asked this question the other day, and the reason would be obvious once we come up with the solution. So look at the solution. So this is how it goes, as it goes quadratically. Essentially, as you see, it increases with radius, right? It increases quadratically with radius. So it's some constant plus another constant times r square. Is it clear? So it's one constant, some a plus b times r square. So it increases quadratically with the radius. Now what you know is that the concentration at the outside, at this interface between the fungus and the air, is a constant, which means, in other words, it decreases quadratically as you go inside the fungus. Is it clear? So what will happen is, just try to envision this, as you keep increasing the size of the mold, what will happen? The gradient will get steeper and steeper, right? Because the concentration that you provide at the outside, that the interface between the mold and the air is a constant. So as your radius keeps increasing, the gradient will get steeper and steeper. Fine. And there will come a point in time. So this is a steady state, under a pseudo steady state approximation we are making this, but remember, this r is actually a function of time. So when you make a pseudo steady state approximation, how do you do that? How do you enforce that? So first you solve the steady. So if I ask you to solve a problem using pseudo steady state approximation, so what is the idea? You decouple the two phenomena. How do you decouple the two phenomena? By first showing that the time scales of these two phenomena are different. So the time scale which is much smaller is under steady state. So you write a steady state equation just like we did and solve for it. Then you solve the unsteady state equation and get how radius varies with time. For example, here if you have your dr dt, if I remember, here, okay, dr dt equals k. Fine. So if this is what you have, then you got your x as a function like this, and you can also integrate dr dt equals k over here right away and get how radius varies with time. So radius here would vary some, linearly. For example, here it's vary, so some kt. Fine. So what you do here is that after you solve for the steady state, you impose that temporal dependence of radius on this equation. Or in other words, if radius, for example, varies linearly with time over here, or radius varies exponentially with time over here, then you go and put that into this equation. Is that clear? Let me write it if it's not clear. So you C naught, the solution that you get here is C naught minus r squared nu max 1 minus r squared. Now if r equals some r naught at t equals 0 times, let's say exponential, I'm just assuming exponential minus kt, something like this. So sorry, it should be plus kt in this case if it's increasing with time. So what you do is you put, so this is your essentially your r at any point of time. So you put that over here, and what you get is 1 minus r naught exponential kt right. So if there's not exponential kt, it's some linear dependence, and that will come in. So this is the way to, is that clear? This is the way to solve a pseudo approximation, when you have a pseudo steady state, or there is a decoupling of two states, then this is the way to solve it. First go and solve for the steady state, and then solve for the unsteady state and impose that unsteady state condition on the steady state solution. Is it clear? This is the way to do it. Fine. Now excuse me. So what you find over here, what you find is that if just by looking at this equation, that as the radius keeps increasing, the concentration gradient becomes steeper. Now the concentration outside is a constant, remember. If the concentration outside were not a constant, if you are increasing it, then no problem. But because the concentration outside the mold, so concentration here is a constant, okay, is a constant, and this radius keeps increasing, what will happen? There will come a point in time when the center of the mold would stop getting any oxygen at all. Is it clear? There will come a point in time when the center of the mold will stop getting any oxygen at all. Okay, and that means that the center of the mold can no longer grow, and that is my critical radius. So let me show you the definition of that. So at critical radius, the critical radius is defined as that radius when the center of the mold is depleted of oxygen, and if the center of the mold is depleted of oxygen, then the growth stops, okay, because the thing has to push from outside, you know. So this is a radius that we want to evaluate, and how would you evaluate that? From this expression, we put r equals, this r equals what? How do we get this r critical? See? Yeah, but that's fine. This side is 0, but there is a variable out here. Critical radius is a constant, and r over here is a variable. So CO2 is equals 0. I agree. What else?
That will happen at the center.
Yeah. So yeah. So this r has to be 0. This r, remember, big R is the external radius. Okay. This radius at r equals 0, the CO2 has to be 0. So we put 0 over here, and you will simply get the critical radius right away as square root of 6 S naught d effective over nu max. So I hope this answers your question from last time, that fungus cannot grow indefinitely, and there will come a point when it will stop growing, and that is the time it will try to divide itself so that it can continue to grow. So this is the limit of the colony. So it's a colony that kind of keeps expanding, but it doesn't expand indefinitely. It expands only up till the time that the radius equals the critical radius. So which is given over here, square root of 6 S naught d effective over nu max. So once this critical radius is attained, then the fungus will stop growing, and it will maybe divide and allow for future growth. Is it clear? So what we looked at is, we're looking at different things at the same time. So first we looked at the normal growth kinetics, and how the kinetics changes, and what is the dynamics of the kinetics, and how different models had been proposed to sort of quantify these dynamics. And what we figured is that of all the kinetic models that have been proposed, it's either the monode model or the modified monode model which are the most useful models. Then we looked at the effect of mass transfer, did a couple of examples to look at the effect of mass transfer, and just as we had seen that in the case of immobilized enzymes, mass transfer affects kinetics, or in other words, kind of decelerates kinetics. Similarly, mass transfer effects here are going to decelerate or slow down the growth process. But there is no running away from it, because for the growth process, just as in the enzymatic process, you need the substrate. You need to provide the oxygen and the nitrogen and the hydrogen and the carbon, just as we showed in the first slide of this whole chapter. You need all of these substrates to be available to the cell, and without this, there is no way you can have cell growth. Now, as soon as you need all of these substrates to be available to the cell, you immediately need mass transfer effect would always mass transfer limitations would automatically come in, so there is no running away from it. So we looked at some of these, and with the effect of for the case of normal growth of single cells and the growth of fungus. So what we said was that the major difference between the growth of cell colony and the growth of fungus is that when a cell colony grows in general, it is like a cell is surrounded by the fluid and therefore by the substrate, and therefore it has direct effect, direct contact with the substrate, whereas a fungus would always grow as a pellet or a mold, or as a lump of colony, and therefore the cells inside the fungus do not have any contact with the nutrients. So the only way the nutrients can come in is through the interface from the outside. It's slowly diffused, and this is the basic difference. And therefore we looked at the mass transfer effects for single cell, and then we looked at the mass transfer effects for the fungus mold. So we are at this point. Now what we're going to do for the rest of the lecture today is look at some other effects, and those effects are the effects of multiple substrates and the effects of inhibition, which mean that if you have more than one substrate, right, so you can provide substrates in different forms and both can lead to cell growth, then what happens, right? So for example, you can give your sugar, both in terms of sucrose and glucose, maybe two different forms, fructose or sucrose or glucose, 2 or 3 different forms. Now all of these substrates, they have different kinetics. Remember, each of them will have different kinetics, but all of them lead to growth of the cells. Fine. So how do we account for that? A. B is that what would be the effect of inhibition, just like we did in the enzyme case. So there, just as you have substrates, there could be inhibitory things that are there. A. And B is that when you give too much of substrate, that one of the things that we talked about, I think earlier, you can have what is known as substrate inhibition. So how do these affect the cell growth process? And we are just going to look at that. I hope we can finish this today. Then we can start a new chapter. But anyway, we're not going to hurry through it. We're going to do it slowly. The first thing we look at is the effect of multiple substrates. Okay. So multiple substrate means that you have cells which are exposed to 2 different substrates, and both these substrates will lead to the product formation, that is daughter cells, but through different kinetic routes. So this is what you see on the screen. Is X is your cell, starting cell, and S1 is substrate number 1, and you get the daughter cell X, 2X, but through the formation of the intermediate compound intermediate cell X prime, which is one route with a set of reaction coefficients K1, K minus 1 and K3. And then you have another substrate that is there parallelly in the system. So you might have given 2 substrates to the system. So that gives again the daughter cells 2X, but through different route X double prime, and the reaction rate constants are K2, K minus 2 and K4. Now I talked about the yield yesterday, if you remember. In the last class I talked about yield, and so the yield is the amount of daughter cells produced per mole of the substrate. Okay, so for case 1, what is the yield? 1 daughter cell is produced from 2 cells. You get 1 cell, you get 2 cells, which means effectively 1 extra cell is being produced per A1 mole of the substrate. So the yield of X over S1 is 1 over A1, and for case 2, the yield of X over S2, that is 1 daughter cell is being produced per A2 moles of S2. So yield is 1 over A2. Fine. Now how do we find, so what we are trying to find is how do we find the overall growth rate of the system? So what is a goal that has to be very clearly defined? So what did we do? We have done only one single substrate before, and we have found the growth rate. So which is dx dt equals mu times x, where mu is given by the monode kinetics, S naught over K plus S naught, some R max S naught over K plus S naught. So here we are trying to find out what would be the form of the overall growth of the system, because what we are essentially interested in at the end of the day is the overall growth. I start with 5 million cells. How many cells do I get after 24 hours or so? Okay, so that is what I'm interested in. Excuse me. So the balance equations, excuse me, the balance equations over here are this. So what I'm writing is I'm writing a balance for the intermediate species, del x prime del t, and as you see over here, the first term is K1 XS1, okay, which is the term because of this forward reaction over here. Okay, see where the arrow is. So this forward reaction over here leads to this term. The second term is minus K minus 1 X prime, which is the result of the backward reaction over here. The first reaction, the backward reaction of that. And the third term over here is minus K3 X prime, which is the result of the formation of the product of the daughter cells. Is it clear? These three terms. It's very straightforward. And then the same thing is repeated for X double prime. Okay, so K2 X times S2 minus K minus 2 X double prime minus K4 X double prime. Is it clear to all? Okay. So, is there a question? No, power, no. Yeah, that, no. Okay, I'll explain that. See, this is not an elementary reaction. See, when do you have the power? Let me explain that if there is any doubt. A plus B giving C, then rate of formation of C is K Ca Cb if it's an elementary reaction. Okay, A plus 2A 2B giving C, the rate is could be given as A Ca Cb square only if and only if it's an elementary reaction. Is it clear? If it's not an elementary reaction, there is nobody said that that's the rate. So it could be this.
When can that not be an elementary reaction?
This one.
No, A plus 2B gives rise to C.
Yeah. Elementary reaction means, the definition of an elementary reaction is that the rate of the reaction is written exactly at the same way in which the molar concentration composition of the reactants react. That is the definition. Okay. See, this reaction, for example, it's defined in elementary reaction. It would be defined only if, so if this molar concentration is 1:2 mole, it reacts in 1:2 mole ratio, and the reaction rate is also given as CaCb square. Okay, but it is not necessary that exactly the mole ratio in which they react will be the ratio in which the rate of the reaction is measured, because the elementary reaction means that there are no other effects apart from the molecular collision between the two molecules that govern the rate of the reaction. Do you understand what I'm saying? So if there are no other effects, so it is, so this is very unlikely that this will be an elementary reaction. Why? Because the probability of two molecules of B and one molecules of A coming and colliding and forming one molecule of C is very, very small. So typically elementary reactions are of the or either first order or second order. Okay. So, when you talk of a first order reaction, so you cannot be an elementary reaction because one molecule cannot combine with itself. There has to be a molecular collision. So what does elementary reaction mean? That the way two molecules collide with each other is exactly the way the reaction is represented as also when the reaction rate is written. So do not assume that whenever you write so this is a molar ratio, this is a stoichiometry. So do not confuse stoichiometry with the power of the reaction. The stoichiometry would be same as the power of the reaction only and only if it's an elementary reaction. Elementary reaction means that all other effects are ignored. So it is directly that the two molecules are combining and that is forming the reaction. But in most cases, it's not like that. There are complex steps out there. For example, this step that I have written over here, as we do in the enzymes, they are not necessarily elementary reactions, which means that it is not that this step alone is occurring. There are multiple steps that are probably occurring over here, and they are summarized as these reversible reactions. Okay, and elementary reaction means that one single step reaction, and in that ratio, so the molar ratio, the stoichiometric ratio that is there, so they combine, the two molecules that combine and form. So this is something very fundamental. You should not make a mistake about it. So if I want to tell you that this is the rate of the reaction, then I will say that okay, this is the reaction, and which reaction 1 is an elementary reaction, which means that in that case you will raise this to the power A1. Okay, but then elementary reaction, if A1 is not an integer, then it cannot be an elementary reaction also. Do you understand what I'm trying to say? If A says 0.75, it cannot be an elementary reaction because 0.75 of a molecule cannot combine with another molecule. So for elementary reaction, A1 has to be a number, typically 1 or at best 2. Even 2 is hard, but at best 2, but not more than 2, because 3 molecules, the probability of 3 molecules combining with each other goes down. If you have read some of statistical mechanics, you will understand that the probability goes down exponentially. So anyhow, no, so this is the reaction rate. And so the next step what we do is we write the constraint equation like we did before, which is the total amount of cells present in the system. So xt is the total amount of cells present in the system. So the total amount of cells present in the system is either in the form of x, okay, which is the daughter of the mother cell, or in the form of any of the intermediate species, x prime or x double prime. Okay, so this is my constraint equation. Next, what I do is use a pseudo steady state approximation or a quasi steady state approximation like we did before, that del del t of x prime equals 0 and del del t of x double prime equals 0, right? So now let me explain this a little bit from what is written in the last line. Okay, so dx dt, so my x equals xt equals x plus x prime plus x double prime. So dx dt would be dxt dt minus dx prime and dt minus dx double prime dt, right? But these are 0 because of pseudo steady state approximation. So you get this as dx dt, right? You get this as dx dt. Now this equals k3 x prime plus k4 x double prime. Can you tell me why is that? Dxt dt equals k3 x prime plus k4 x double prime. Right. So xt is what? Xt is the overall amount of cells that are present. Now, see, the only difference here from what we did in the enzyme case, if I'm allowed to go back to the slide. So look at this. So this is the reaction that is occurring because of the formation of the daughter cells that are occurring through the from x prime or x double prime to 2x. Okay. So what is the major difference from what we're doing here from what we're doing in the constraint? This xt is not a constant. That's the only difference. In the enzyme case and all the previous cases we had done, this xt was a constant, right? But here the xt is not a constant. So that is a major difference that is there. Why? Because there is an overall growth of the cells. And how is this overall growth quantified? If I come here, so what is the overall growth? If you look at the system, for example, the overall growth is one daughter cell growth from this. Okay. So the total growth for the system dxt dt, ddt of xt equals, so k3 times x prime plus k4 times x double prime. Okay. So one daughter cell growth each of these reactions. First reaction, one daughter cell growth, and second reaction, one daughter cell growth. Is it clear to everybody? Okay, this is a little tricky part. Okay, so this is what we have. So I hope you agree with me. Now dx prime dt equals 0. So x prime equals, if you go back to your notes and check the, I can go back to the slide in a minute, but just let me write this. This is what I get. If you go back to your nodes and the pseudo steady state, this is a pseudo steady state approximation. Pseudo steady state approximation. This is what we meant. So you will see that dx prime dt equals 0 would give you this. Okay, and if you have a doubt, I'll go back to it in the here. So look at this. So what you have is k1 x s1 minus k minus 1 x prime minus k3 x prime. Okay. So from here you can, if this side, the left hand side is 0, you can straight away express your x prime as a function of x and s1. Clear to all of you? The same thing you can do over here with x double prime. If your ddt of x double prime is 0, then you can express your x double prime as a function of x and s2, right? So this is what we do over here. So ddt of x prime is written as 0 and x prime is given by this number, and x double prime is written as this number. So why are we doing it? Because we intend to substitute these two into our constraint equation, just like we had done before. Okay. So when we substitute these two into our constraint equation is here at the top, and what we do is we substitute these two into our constraint equation. So this is what I get. So xt equals x times, so x is the first term, then x prime here. So you take x out of the parenthesis, common out of the parenthesis. So you get this and get this. But the only thing that you have to keep in mind again, that xt is not a constant over here, it is varying with time. So that is a major difference between what we are doing now to what we did in the previous constraint equation. So now we can take a derivative of this. Okay, I'll continue as soon as you finish writing. Okay, shall I go ahead? Okay. So now we take the derivative. So dx, this was already something that I had written. Dx dt, ddt of xt is k3 x prime plus k4 x double prime. So you do the same thing again. You replace your x prime by what you got from the pseudo steady state approximation. You replace your x double prime by what you got from the pseudo steady state approximation. Fine. Just write the first step, then I'll show the next step. Just write the first line, then I'll show you the how to get the second line. You probably know that already, but I'll still show you. So dx dt equals k3 x prime plus k4 x double prime. So this you replace, as soon as you replace, you get x times this number k3. Fine. Now what you do is, let's go to the slide. So here you have the here the last line, you have the relationship between xt and x. Okay. So because, why are you doing this? Because we want to find everything in terms of the total amount of cell that is present in the system. So because it doesn't matter to us which one is in the pseudo steady state, which is the daughter, which is the, because what you can measure essentially at the end of the day is the total amount of cells. You under a microscope you cannot differentiate that this is in the intermediate species is the final and this is the initial species. So what you can measure is the total amount of cell. So that is why all our calculations we want to do it in terms of the total amount of cells. So xt over here is 1 plus k1 is 1, k minus 1 k3 plus k2 is 1, k minus 2 plus k4. Fine. So if this quantity, let me, it's too big, so let me call it this b or something. So if x is xt times b inverse, okay, then you can put it over here into this equation, this whole thing. Okay. So xt is so then you can get it in that form. And let's call this thing a. Okay, this term is a, this term is b. Then what you have is, so what I did was I just said this term is a, this term is b, and then what you have is dx dt equals xt times a times b inverse. Right? Now what is my mu? Mu is a specific growth rate. Fine. How is it defined? 1 over xt times dxt dt, like the first order rate constant. Clear? So from this, what do I get from this equation? From here, what I get, this equals A times B inverse. So I got my specific growth rate. Now the specific growth rate for the monode model, if you remember, was some S, Ks times Rx, Rmax S times K over K plus S, right? That was the monode growth model, mu. Right? So now you can, so this is what you get. So exactly the, once we do the steps that we did on paper, this is what you get. And mu is your new new rate, specific growth rate. So once you do the simplification, which we are not doing because of lack of time. So this is your mu, this inverse time this, and there's, if you multiply both of these, and you can do a little bit of simplification, then you find out that mu comes out to be in a very nice compact form. And what is interesting over here is that that's the thing to note, is that the substrates influence each other. So the specific growth rate of the two substrates put together is not equal to the specific growth rate of substrate 1 and plus the specific growth rate of substrate 2. They are not independent. The substrate, the two growths are not independent of each other. And what you find over here is that this is specific growth rate of substrate 1, but it is being influenced in the presence of substrate 2. There is an influence out here, alpha 2s2. Similarly, the specific growth rate of substrate 2 is being influenced by the specific growth rate of substrate 1, and there is an influence out here in terms of alpha 1 s1. Okay. And alpha 1 and alpha 2 are constants, obviously. So again, when you come to alpha 1 and alpha 2, interestingly, you will see that alpha 2 and alpha 1, both of them involve all the rate constants of the reactions. So K1, K minus 1, K3, K2, K minus 2, K4. So all the 6 rate constants of the two reactions are involved in alpha 1 and alpha 2. And not surprisingly, what you find that mu max 1 equals K3 and mu max 2 equals K4. Why is that not surprising? Because the maximum rate possible is the one, in the absence of everything, just the straightforward production rate. And why I'm saying that's interesting is because, we didn't presume anything. We just went ahead and did the calculations. And when you got the final results, you can make some physical sense out of those results. So K1 is the specific, the rate constants involved in the reaction 1. K2 are the rate constants involved in reaction 2. And as I said, that the mu max, maximum growth rate of reaction 1 is K3. The maximum growth rate of reaction 2 is K4. Fine. So this is an important result, and you might want to at least, if not remember it completely, at least have a sense of what's going on. And this is not difficult to remember actually, because this is a monode growth kinetics, and you just add one term in the denominator, and this is a monode growth kinetics, add one term in the denominator. What are difficult to remember might be these constants. So those might be difficult to remember. So the next thing we do is the effect of inhibition. So here, first thing we looked at is the effect of multiple substrates, but the next thing that we do is the effect of inhibition. So what if the substrate, if you have one single substrate, but instead of the substrate, along with the substrate producing the cells, the substrate inhibits. So this is exactly the same, if you remember, as substrate inhibition that we did for the case of enzyme kinetics, exactly the same. So the kinetics is also the same. So in there also we had the thing X plus S giving XS, which in earlier case gave products, but in this case our product is the new cells. So X plus S giving XS gives new cells, and this is the inhibition. So X plus S, more substrate if that's present over there. Instead of giving new cells, it gives this complex XS2, which does not lead to product formation. Okay, so we have to go through a similar set of steps that we did for inhibition in case of enzyme kinetics, except that in this case the difference being that this is varying with time. So xt is not a constant, but xt is varying with time. Okay, so xt of dxt ddt of xt over here is k times xs, right? Is that clear to everybody? Why is that? Because this second reaction is not leading to any growth of cells, so so that is not considered over there. So ddt of xt is k times s. Now k times xs, sorry. Now from here, from the first reaction, the reversible first reaction, what you find is, if this is assumed to have attained equilibrium, then xs could be written as x times s could be written as ks times xs. Is it clear to everybody? Very straightforward. So we can replace my xs that I have over here by x times s divided by ks. Right? So you have k over ks times x times s. Okay. Now the other thing is to take care of this xs too. So my constraint equation again is the total amount of cells that is present. Cells are present as parent cells and daughter cells as x, in terms of the complex, intermediate complex xs, and in terms of the complex that you have is xs2. So the cells are present in these three different forms. Fine. Again, I want to reiterate that xt is not a constant, but it varies with time. So ddt of xt is not 0, but what we do over here is that from this inhibition reaction, the substrate inhibition reaction, this we again assume to have attained equilibrium. Fine. We had assumed the substrate inhibition reaction given here to have attained equilibrium, and as a result we can write ki as xs times xs times xs2. Clear? Then xs again we can write from this equation as x times s over ks. Clear? So that is what we do. So we write this x plus xs plus xs2. So x plus xs I have substituted from here. So x times s over ks, and xs2 I have substituted from here. So xs2 is from here is xs times s over ki. Fine. Where xs could again be substituted by x times s over ks. Right. So I can write this like this, and then then I can take x common out of the whole thing, and this is the part I get. I'll just show you if there's any problem with this. So xt equals x plus xs plus xs2. I'm just doing away with the brackets. So xs2 equals xs times s over ki. Now xs equals, this is from the x times s over ks. I replace this over here. So what I get is x times s square over ki ks. So from here, if I replace it over here, I get x plus xs x times s over ks plus x times s square over ki ks. So you can take it out. So what you get over here is that dx dt equals ks k over ks times x times s. The reason being that this is the final reaction. Okay, this is the only thing that generates products. Fine. Is it clear? So this is the only step that generates products. So you have k over ks times xs, and then you can substitute your x in terms of xt that we got. So x in terms of xt we got over here. You substitute that back from here, and you get in terms of xt. So now my specific growth rate is 1 over xt ddt of xt. So what is that? That is now my mu over here. Right? So what you find is mu is this. So what is the difference? The major difference that you find over here is as compared to the mu that you have before, was that the mu before had something which is similar to a monod growth kinetics, but this is no way similar to the close to the monod growth kinetics. And if you remember what we did from last time, what is going to be the characteristic of the mu? How is this, how is this mu going to be different from the mu's in the last cases? Graphically, let's think graphically. So mu over s in monod growth kinetics is going to be something like this, right? So monod So, for multiple you will have S1 and S2. Okay. So, say mu1 is something like this, mu2 is something like this. For multiple, this is mu. So, how is this one going to vary? Yeah, so it will go through a maximum and then go down to 0. So, this one starts at 0 and saturates. This one starts at 0 and goes to 0 through a maximum. Okay. Why is that? If you look at the expression itself, you will see that. Okay, that it will saturate out in the absence. So, how do you obtain the maximum? Just simply differentiate it and do a del mu del s and get that. So, we did a del mu del s to get the maximum value, and the maximum specific growth rate turns out to be S critical is square root of KIKS. Okay. Square root of KIKS. It's similar, if you remember from last time, in the case of enzymatic growth, it's exactly similar to what we did in the last case. Okay. When you're done, we'll move on to the last thing. And you could, for the last case, you can do some calculations on your own also. So, this is a mu, just do a del mu del s and you get this. Okay. So, shall we move on? Okay. So, the last thing that we are going to do is in today's lecture is allosteric inhibition, and this is the last thing that we are going to do in this chapter also. So, in this kind of inhibition, what happens is that instead of stopping here, see what is the difference between the last case and here is that last time we stopped here, XS2, and we stopped here. In allosteric inhibition, what happens is that this also gives rise to daughter cells. So, we do not stop here completely, we say that this also gives a rise to daughter cells. So, in a way, it is a combination of substrate inhibition and multiple substrates. Come to think of it, do you agree with me? So, in a way, it's a combination of substrate inhibition. In substrate inhibition, we had everything like we had, but we stopped over here, right? There's no daughter cells produced from the second reaction. But in the multiple substrate, we didn't have the same substrate combining, but we had daughter cells from both the substrates. So, in a way, it's like a combination of multiple substrates and substrate inhibition. So, I wouldn't do all the steps for it, I'll do some steps, and you can go ahead and do the rest either here or later, maybe at home as an assignment. Okay. So, the procedure is that the same procedure as before. The only difference that is going to happen is that del del t of x t is k2 times xs plus this beta k2 or k3, k4 also we can call it, but we just call it beta k2 just for the sake of algebra, times xs2. Is that clear? So, I think that there is nothing hard in it. So, and then you have to go and do the rest of the algebra, which is the xt you have to, you know, calculate the xt, which is the x plus xs plus xs s as before, and do the del del t of xt, and then take the reversibles for both cases, and this is the final result that you will come up with for mu. Mu is defined as 1 over xt del ddt of xt, and this is the final result you will come up with. So, the difference between the difference between last time and this time is that last time you had the denominator and you had one term in the numerator, you have another term in the numerator. So it's quadratic both in the numerator and the denominator, and when you again it will have a lot more interesting kinetics. So what I would like you to do, this is probably my last slide here. So what I'd like you to do is actually go and plot this, because this is not necessarily going to have the same kinetics as that, and it can even have multiple peaks. I'm not sure, but once you differentiate it, you'll be able to figure out that if it has multiple peaks. So what I want you to do as a little assignment is that do the steps. The steps are very straightforward, but do the steps and check the answer. And then once you've got this answer, I want you to plot this A, and B, C. The first thing you should actually do is go and differentiate it to find the maxima and the minima, because once you get the maxima and the minima, you can plot it very straightforward. Otherwise, it's like a black box trying to plot this. So go and find the maxima and the minima. One of the maxima is going to be like what we had before, okay, but then there's a quadratic term, so it could lead to some other interesting effects. So I want you to do that. So this is something very common that happens, and this is the specific growth rate in that case, and we need to understand how this works in order to be able to figure out. So the reason we are doing this is what we're going to do in the next chapter, and all these cases that we did today are important, because I'm going to quiz you on those things later in the exams. So in the next chapter, what we're going to look at is the process, the dynamics of cell growth. See, we are looking at the kinetics of cell growth, okay, but what happens is that the cell growth process happens in a reactor like this, so it could be a batch reactor, it could be a chemostat, a continuous tank reactor. So one of the things that we are going to look at is, starting next class, it's a new chapter, is chemostat, which is a CSTR for a biological reaction. So temperature is controlled, and pH is controlled, so pH and temperature controlled, and then we look at the growth process inside this reactor, okay. So that is when all the kinetics that we are doing in this class and the previous classes and the mass transfer effects and so on come into play. So right now we're looking at, so this is all connected. So right now we're looking at the kinetics in itself, just isolating the kinetics and looking at it. Then we take the kinetics out and put it into the reactor and look at what the effects are. And these are, I assure you that some of these effects are going to be very interesting. So we will work with, so probably what we'll start with is not even this, the very straightforward case here, the monode kinetics, and then we'll go and slowly do what I'll probably ask you to do, what the effects of multiple reactions are, the effects of inhibition, and the effects of allosteric inhibition. So we'll stop here, and I'll see you in the next class. Thanks.